The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
All right, we should probably get started. So RNA plays important regulatory and catalytic roles in biology, and so it's important to understand its function. And so that's going to be the main theme of today's lecture. But before we get to that, I wanted to briefly review what we went over last time. So we talked about hidden Markov models, some of the terminology, thinking of them as generative models, terminology of the different types of parameters, the initiation probabilities and transition probabilities and so forth. And the Viterbi algorithm, which is sort of the core algorithm used whenever you apply HMMs. Essentially, you always use the Viterbi algorithm. And then we gave as an example the CPG island HMM, which is admittedly a bit of a toy example. It's probably not really used in practice, but illustrates the principles. And then today we're going to talk about a couple of real-world HMMs. But before we get to that, I just wanted to sort of toward the end, we talked about the computational complexity of the algorithm and concluded that if you have a k-state HMM run on a sequence of length L, it's order k squared L. Okay? And this diagram is helpful to many people in sort of thinking about that. So you can have transitions from any state, for example, from this state to any of the other 5 states in this 5-state HMM. And when you're doing the Viterbi, you have to maximize over the 5 possible input transitions into each state. And so the full set of computations that you have to do from going from position I to I+1 is k squared. Does that make sense? And then there's L different transitions you have to do, so it's k squared L. Any questions about that? All right, and so the example that we gave is shown here. And what we did was to take an example sort of where you could sort of see the answer. Not immediately see it, but if you're thinking about it a little bit, figure out the answer. And then we talked about how the Viterbi algorithm actually works and why it makes the transitions at the right place. It seems to intuitively like it would make a transition later, but actually it transitions at the right place. And one way to think about that is that it's not making, these are not hard and fast decisions. When it's making, because you're optimizing 2 different paths at every state, you're considering 2 possibilities. And so you explore the possibility of, the first time you hit a C, you explore the possibility of transitioning from genome to island, but you're not confirming whether you're going to do that yet until you get to the end and see whether that path ends up having a higher probability at the end of the sequence than the alternative. Okay, so that's sort of one way of thinking about that. Any questions about this sort of thing? How to sort of understand when a transition will be made? And I want to emphasize, for this simple HMM that we talked about, you can kind of see what the answer is going to be. But if you have any HMM, any sort of interesting real-world HMM with multiple states, there's no way you're going to be able to see it. I mean, maybe you could guess what the answer might be, but you're not going to be able to be confident of what that is, which is why you have to actually implement it. Okay. All right, good. All right, let's talk about a couple of real-world HMMs. So I mentioned gene finding. That's been a popular application of HMMs, both in prokaryotes and eukaryotes. There's some examples discussed in the text. Another very popular application are so-called profile HMMs. And so this is a hidden Markov model that's made based on a multiple alignment of proteins which have a related function or share a common domain. For example, there's a database called PFAM, which includes profile HMMs for hundreds of different types of protein domains. And so once you have many dozens or hundreds or thousands of examples of a protein domain, you can learn lots of things about it. Not just what the frequencies of each residue are at each position, but how likely you are to have an insertion at each position. And if you do have an insertion, what types of amino acid residues are likely to be inserted at that position, and how often you are likely to have a deletion at each position in the multiple alignment. And so the challenge then is to take a query protein and to thread it through all of these profile HMMs and ask, does it have a significant match to any of them? And so that's basically how PFAM works. And the nice thing about HMMs is that they allow you to, if you want to have the same probability of insertion at each position in your multiple alignment, you can do that. But if you have enough data to observe that there's a 5-fold higher likelihood of having an insertion at position 3 in the multiple alignment than there is at position 2, you can put that in. You just change those probabilities. Okay, so in this HMM, each of the hidden states is either an M state, which is a match state, or an I state or an insert state, and so those will emit actual amino acid residues, and or it can be a delete state, which is thought of as emitting a dash, a placeholder in the multiple alignment. Okay, so these are also widely used, and then one of my favorite examples, it's fairly simple, but turns out to be quite useful, is the so-called TMHMM for prediction of transmembrane helices in proteins. So we know that many, especially eukaryotic proteins, are embedded in membranes, and often, there's one famous family of 7 transmembrane helix proteins, and there are others that have 1 or a few transmembrane helices. And knowing that a protein has at least 1 transmembrane helix is very useful in terms of predicting its function. You predict its localization. And knowing that it's a 7 transmembrane helix protein is also useful. And so you want to predict whether the protein has transmembrane helices and what their orientation is. That is, proteins can have their N-terminus either inside the cell or outside the cell. And then, of course, where exactly those helices are. And this program has about a 97% accuracy, according to the author. So it works very well. So what properties do you think? We said before that you have to have strongly different emission probabilities in the different hidden states to have a chance of being able to predict things accurately. So what properties do you think are captured in a model of transmembrane helices? What types of emission probabilities would you have for the different states in this model? Anyone? So, like, taking for this protein, like, what kinds of residues would you have in here? Oops, sorry. I'm having trouble with this thing. Okay, right here in the middle of the membrane, what kind of residues are you going to see there? Those are going to be hydrophobic, exactly. And what about right where the helix emerges from the membrane? Often you see charged residues there that kind of anchor it, that prevent it from sliding back into the membrane. And then in general, both on the exterior and interior, you'll tend to have more hydrophilic residues. Right? So that's sort of the basis of TMHMM. And it also, so this is the structure, and you'll notice that these are not exactly the hidden states that correspond to individual amino acid residues. This is sort of these are like meta states, just to illustrate the overall structure. I'll show you the actual states on the next slide, but these were the types of states that the author, Anders Krogh, decided to model. So he has sort of focuses here on the helix core, okay? There's also a cytoplasmic cap and a non-cytoplasmic cap. Oops, didn't mean that. And then there's sort of a globular domain on each side, both on the cytoplasmic side, or you can have one on the non-cytoplasmic side. Okay, so there's going to be different compositions in each of these regions. Now, one of the things we talked about with HMMs is that if you were, now let's think about the helix core. The simplest model you might think of would be to have sort of a helix state, and then to allow that state to recur to itself. Okay, so this type of thing where you then had some transition to some sort of cap state after. This would allow you to model helices of any length, right? But now, how long are transmembrane helices? What does that distribution look like? Does anyone have an idea? There's a certain physical dimension, right? Phospholipids, it takes a certain number of residues to get across here, right? And then that number is about 20-ish, okay? So transmembrane helices tend to be sort of on the order of 20 plus or minus a few. And so it's totally unrealistic to have a transmembrane helix that's like 5 residues long, right? So if you run this algorithm in generative mode, what distribution of helix lengths will you produce? We're running it in generative mode where we're going to let it, remember, generate a series of hidden states and then associated amino acid sequences. It's coming from some, let's say, I don't know, what kind of states are there here? Caps, plasmid. Let's say, goes into helix, hangs out here. I'm sorry, is there an answer to this question? Anyone? I don't know how long. If I let it run, it'll generate a random number. It depends on what this probability is here. Let's call this probability p, and then this would be 1 minus p. Okay, so obviously if 1 minus p is bigger, it'll tend to produce longer helices, right? But in general, what is the shape of the distribution of consecutive helical states that this model will generate?
Binomial.
Binomial? Okay, can you explain what?
Because the helix would have to have the helix of length n would occur like 1 minus p to the n power.
Okay, so a helix of length n, so the probability that, say, let's call it L, the length of a helix, equals n is 1 minus p to the n, right? Is that binomial? Someone else? Yes?
Is it negative binomial?
Negative binomial, okay.
So the number of times it stays in the helix state before moving out, it's not that.
Yeah, so the distribution is going to be like that, right? You have to stay in here for n and then leave, right? So this is, the simplest, you could have special cases of binomial and negative binomial, but in general, this distribution is called the geometric distribution, or the continuous version would be the exponential distribution, right? So what is the shape of this distribution? If I were to plot, n down here on this axis, and the probability that L equals n on this axis, what kind of shape can someone draw in the air? So you had like up and then down? Okay, so actually it's going to be just down, right? Like that, right? Because as n increases, this goes down, right? Because 1 minus p is less than 1, right? So it just steadily goes down. And what is the mean of this distribution? Anyone remember this? Yeah, so there's sort of two versions of this that you'll see. One of them is the 1 minus p n minus 1 p, and one of them is this. And so this is the number of failures before a success, if you will, if success is leaving the helix. And this is the number of trials till the first success, right? So one of them has a mean that's 1 over p, and the other has a mean that's 1 minus p over p. Okay. So usually p is small, and so those are about the same. Okay, so 1 over p, you can think about it, 1 over p is roughly right. And so if we were to model transmembrane helices, and if transmembrane helices are about, I said about 20 residues long, you would set p to what value? To get the right mean.
0.05.
Yeah. 0.05 1/20th, so that 1 over that will be about 20, right? Okay. So, and then 1 minus p would, of course, be 0.95. Okay. So if I were to do that, I would get a distribution that looks about like this, with a mean of 20, okay? But if I were to then look at real transmembrane helices and look at their distribution, I would see something totally different. It would probably look like that, right? It would have a mean around 20, but the probability of anything less than 15 would be 0, right? That's too short. It can't go across the membrane, right? So, and then again, you don't have ones that are 40. They don't kind of wiggle around in there and then come out. They tend to just go straight across. So yeah, so there's a problem here. You can see that if you want to make a more accurate model, you want to capture not only get the right emission probabilities with the right probabilities of hydrophobics and hydrophilics in the different states, but you also want to get the length right. Okay, and so the trick that well, actually, yeah, can anyone think of tricks to get the right length distribution here? How do we do better than this? We're basically hidden Markov models where you have a state that recurs to itself. It will always be a geometric distribution. The only choice you have is what is that probability, and that will give you, so you can get any mean you want, but you always get this shape, right? So if you want a more general shape, what are some tricks that you could do? How could you change the model? Any ideas? Yeah, go ahead.
In your Markov model, you have multiple helix states.
Multiple helix states. Okay. How many?
Proportional to the length you want, the sequence to.
Like 1 for each possible length. Is that
Maybe less than 1.
Or less than 1, right. So you could have something like, I mean, let's say you have like this. Helix begin, or I don't know, helix 1, helix 2. You allow each of these to recur to themselves, okay? What does that get you? This actually gets you something a little bit better. It gives you a little bit of, it's more like that, okay? So that's better, but if I want to get an exact distribution, then actually so this is the solution that the authors actually used. They made essentially 25 different helix states, and then they allowed various different transitions here. So by setting, it's a little arbitrary here, but they have this special state 3 that can kind of take a jump. So it can just continue on to 4, and that'll make your maximum length helix core, right? Or it can skip 1, go to 5, and that'll make a helix core that's 1 residue shorter than that, or it can skip 2, and so forth. And you can set any probabilities you want on these transitions, and so you can fit basically an arbitrary distribution within a fixed range of lengths that's determined by how many states you have. Okay? So they really wanted to get the length distribution right, and that's what they did. What's the cost of this? What's the downside? Simona?
I was just going to ask, it looks like from this that your minimum helix length could be 4.
Yeah, that's a good question. Yeah, I think, yeah, we don't know what the probabilities, they said on that. What did they really mean that? And also that's only the core, and maybe these cap things can be able to, yeah, that seems a little short to me. So yeah, I agree. Yeah, I'm not sure. It could just be for the sake of illustration, but they don't actually use those. Yeah. But anyway, the point, yeah, that's a, I mean, probably have to read the paper. I haven't read this paper for many years, so I don't remember exactly the answer to that. But I have a citation. You can look it up if you're curious. But the main point I wanted to make with this is just that by setting an arbitrary number of states and putting in possible transitions between them, you can actually construct any length distribution you want. But there is a downside, and what is that downside?
Computational cost.
Yeah, computational cost. Instead of having 1 helix state, now we've got 25 or something. So and the time goes up by the square of the number of states. So it's going to run slower. And you also have to estimate all these parameters. Right? So, okay, good. So here's an example of the output of the TMHMM program for a mouse chloride channel gene, CLC6. So the program predicts that there are 7 transmembrane helices, as shown by these little red blocks here. You can see they're all about the same, about 20 or so, and that the program starts outside and ends inside. Okay? So let's say you were going to do some experiments on this protein to test this prediction. So one of the types of experiments people do is they put some sort of modifiable or modified residue into one of the spaces between the transmembrane helices, and then you can test, can we modify, by modifying the cells with something that's a non-permeable, chemical, can you modify that protein, right? So only if that stretches on the outside of the protein will you be able to, on the outside of the cell, will you be able to predict it. So that's a way of testing the topology. So if you were doing those types of experiments, you might actually, like maybe you're not sure if every transmembrane helix is correct, right? There could be some where the boundaries were a little off, or even a wrong helix. And so one of the things that you often want with a prediction is not only to know what is the optimal or most likely prediction, but also how confident is the algorithm in each of the parts of its prediction. How confident is it in the location of transmembrane helix 3, or the probability that actually there is a transmembrane helix 3. Okay? And so the way that this program does that is using something called the forward-backward algorithm. Okay, so those of you who read the Rabiner tutorial, it's described pretty well there. The basic idea is that I mentioned that this PO, the probability of the observable sequence, summing over all possible HMM structures, or all possible sequences of hidden states, that is possible to calculate. And the way that you do it is you run an algorithm that's similar to the Viterbi, but instead of taking the maximum entering each hidden state at intermediate positions, you sum those inputs. Okay? So you just do the sum at every point, and it turns out that will calculate the sum of the 2 values at the end, or the k values at the end, will be equal to the sum of the probabilities of generating the observable sequence over all possible sequences of hidden states. Okay, so that's useful. And then you can also run it backwards. There's no reason it has to be only going in one direction. And so what you do is you run these sort of summing versions of the Viterbi in both the forward direction and also run one in the backward direction. And then you take a particular position here, like let's say this is your helix state, for example. So, and we're interested in this position, somewhere in the middle of the protein. We want to know, is that a helix or not? And so basically you take the value that you get here from the forward, in your forward algorithm, and the value that you get here in the backward algorithm, and multiply those 2 together and divide by this PO, and that gives you the probability. Okay, so that ends up being equivalent, a way of calculating the sum of all the parses that go through this particular position I in the sequence in that particular hidden state. Okay. So, I mean, I realize that may not have been totally clear, and I don't want to take more time to totally go into it, but it is pretty well described in Rabiner, and I'll just give you an example. So if you're motivated, please take a look at that, and if you have further questions, I'd be happy to discuss during office hours next week. And this is what it looks like for this particular protein. So you get something called the posterior probability, which is the sum of the probabilities of all the parses, and they've plotted it for the particular state that is in the Viterbi path, that is in the optimal parse. So, for example, in blue here. So the blue is the probability. Well, actually, they've done it for all the different states here. So blue is the probability that you're outside. Okay. So it's very, very confident that the N-terminus of the protein is outside the cell. It's very, very confident in the locations of transmembrane helices 1 and 2. It actually, more often than not, thinks there's actually a third helix right here. Okay. But that didn't make it in the optimal parse. That actually occurs in a majority of parses, but not in the optimal one. Okay? And it's probably because it would then cause other things to be flipped later on if you had a transmembrane helix there. It's not sure whether there's a helix there or not, but then it's confident in this one. Okay, so this gives you an idea. Now, if you were going to, if you wanted to do some sort of test of the prediction, you want to test probably first the higher confidence predictions. So you might, do something right here. Or if maybe you from experience, you know that when it has, a probability that's that high, it's always right. So there's no point testing it. So you should test one of these kind of less confident regions. So this actually makes the prediction much more useful to have, some degree of confidence assigned to each part of the prediction. Okay. All right. So for the remainder of today, I want to turn to the topic of RNA secondary structure. So at the beginning, we'll sort of do some nomenclature, and then to motivate the topic, give some biological examples of RNA structure, gives me an excuse to show some pretty pictures of structure. And then we'll talk about two approaches, which are two of the most widely used approaches toward predicting structure. So using evolution to predict structure by method of covariation, and then which works well when you have many homologous sequences, and then using sort of first principles thermodynamics to predict secondary structure by energy minimization, where obviously you don't need to have a homologous sequence present. And the Nature Biotechnology primer on RNA folding that I recommended is a good intro to the energy minimization approach. Okay, so what is RNA secondary structure? So you all know that RNAs, like proteins, have a three-dimensional tertiary folded structure that in many cases determines their function. But there's also sort of a simpler representation of this structure where you just describe which pairs of bases are hydrogen bonded to one another. Okay? And so for RNA, so it's a famous example of an RNA structure, this sort of cloverleaf structure that all tRNAs have. The secondary structure of the tRNA is the set of base pairs. So it's this base pair here between the first base and this one toward the end, and then this base pair here, and so forth. And so if you specify all those base pairs, then you can then draw a picture like this, which gives you a good idea of what parts of the RNA molecule are accessible. So for example, it won't tell you where the anticodon loop is, which is sort of the business end of the tRNA, but it narrows it down to like 3 possibilities. You might consider that, or that, or down here. It's unlikely to be something in here because these bases are already paired. They can't pair to the message. So it gives you sort of a first approximation toward the 3D structure, and so it's quite useful. So how do we represent secondary structures? So it's a few different common representations that you'll see. So one is, and this is sort of a computer-friendly but not terribly human-friendly representation, I would say, is this sort of dot in parentheses notation here. So the dot is an unpaired base, and the parenthesis is a paired base. And how do you know chalk is sort of non-uniformly distributed here? So if you if you have a structure like this, and you have these 3 parentheses, what are they paired to? Well, you don't know yet, okay, until you get further down, and then each left parenthesis has to have a right parenthesis somewhere, okay? So now if we see this, then we know that there are 2 unpaired bases here, and then there's going to be 3 in a row that are paired, okay, these guys. We don't know what they're paired to yet, okay? Then there's going to be a 5 base pair loop, maybe a little pentagon type thing. 2, 3, 4, oops, 4, 5, okay. And this one would be this, then the right parentheses that pair with the left parentheses over here, okay? I should probably draw this coming out so to make it clearer that it's not paired. Okay, so This notation you can convert to this. So after a while, it gets relatively easy to do this, except when they're super long. Okay. So that's what the left part of that would look like. So what about the right part? So the right part, we have something like 1, 2, 3, 4, a bunch of dots, and then we have 2, and then a dot, and then 2. What does that thing look like? So that's going to look like 4 bases here in a stem, big loop, and then there's going to be 2 bases that are paired, and then a bulge, and then 2 more that are paired. Okay? These things happen in real structures. Okay, and then the arc notation is a little more human-friendly. It actually draws an arc between each pair of bases that are hydrogen bonded. Okay, so I'm sure you can imagine what those structures would look like. Okay, and it turns out that the arcs are very important. Like whether those arcs cross each other or not is sort of a fundamental classification of RNA secondary structures into the ones that are tractable and the ones that are really difficult. All right, so pretty pictures of RNA. So this is a lower resolution cryo-EM structure of a bacterial ribosome. So remember, ribosomes have 2 subunits, a large subunit, 50S, and a small subunit, 30S. And if you crack it open, okay, so you basically split, you sort of break the ribosome like that, and you look inside, they're full of tRNAs, okay? So there are 3 sort of pockets that are normally distinguished within ribosomes. The A site, this is the site where the tRNA enters that's going to add a new amino acid to the growing peptide chain. The P site, which is this tRNA will have attached to it the actual growing peptide, and then the exit tunnel where this tRNA will eventually the E site where which is the one that was added a couple of residues ago. Okay, so one thing people often think of RNA structure just in terms of these secondary structures because they're so they're much easier to generate than tertiary structures, and they give you, like for tRNA, it gives you a pretty good some pretty good information about how it works. But for a large and complex structure like the ribosome, it turns out that RNA is actually not bad at building complex structures. I would say it's not as good as protein, but it is capable of constructing like something like a long tube. And in fact, in the ribosome, you find such a long tube right here that is where the peptide that's been synthesized exits the ribosome. And you'll notice it's not like a large cavity in which the protein might start folding. It's a skinny tube that is thin enough that the polypeptide has to remain linear, cannot start folding back on itself. Okay, so you sort of extrude the protein in a linear, unfolded conformation and let it fold outside of the ribosome. Okay, if it could fold inside, that might clog it up. I think that would be that's probably one reason why why it's not designed that way. Okay. I'm sure that was tried by evolution and rejected. Okay, so if you look at the ribosome, now remember the ribosome is composed of both RNA and protein, you'll see that it's much more of one than the other, right? And so it's really much more of the fettuccine, which is the RNA part, than the linguine of the protein. And if you also look at the distribution of the proteins on the ribosome, you'll see that they're not in the core. They're kind of decorated around the edges. It really looks like something that was originally made out of RNA, and then you sort of added proteins as accessories later, and that's probably what happened. Okay, so if you then, this is based on the structures that were solved a few years ago. If you then look at where the nearest proteins are to the active site, the actual catalytic site. Remember, the ribosome catalyzes peptide, addition of an amino acid to a growing peptide, so peptide bond formation. You'll find that the nearest proteins are around 18 to 20 angstroms away. Okay, and this is too far to do any chemistry. So this structure the active site residues need to be, or molecules need to be, within a few angstroms to do any useful chemistry. And so this basically proves that the ribosome is a ribozyme. That is, it's an RNA enzyme. RNA is doing the catalysis. Okay. All right. Good. So here is the structure of a ribosome. It's very kind of beautiful, and it's impressive that somebody could actually solve the structure of something this big. But what is actually the practical use of this structure? It turns out there's quite an important practical application of knowing this structure. Any ideas?
Antibiotics.
Antibiotics, exactly. So many antibiotics work by taking advantages of differences between the prokaryotic ribosome structure and the eukaryotic ribosome structure. So if you could make a small molecule, these are some examples, that will inhibit prokaryotic ribosomes, but hopefully not inhibit eukaryotic ribosomes, then you can kill bacteria that might be infecting you. Okay, good. All right. Okay. So non-coding RNAs. So there's many different families of non-coding RNAs, and I'm gonna list some in a moment, and I'm gonna actually challenge you, see if you can come up with any more families of non-coding RNAs. But they're receiving increasing interest, I would say, ever since microRNAs were discovered. Sort of a boom in looking at different types of non-coding RNAs. Link RNAs also important and interesting, as well as many of the classical RNAs like tRNAs and rRNAs and SNO-RNAs. There may be new aspects of their regulation and function that will be interesting. And so when you're studying a non-coding RNA, it's very, very helpful to know its structure. If it's going to base pair in trans with some other RNA, as tRNAs do, as microRNAs do, for example, and snRNAs and SNO-RNAs, then you want to know which parts of the molecule are free and which are internally base paired. And if you want to predict non-coding RNA genes in a genome, you may want to look for regions that are under selection for conservation of RNA structure, okay? For conservation of the potential to base pair at some distance. If you see that, it's much more likely that that region of the genome encodes a non-coding RNA than that it codes, for example, that it's a coding exon, or that it's a transcription factor binding site or something like that that functions at the DNA level. And so, sort of having this notion of structure, even just secondary structure, is helpful for that application as well, and predicting functions, as I mentioned. Okay, so covariation. So let's take a look at these sequences. So imagine you've discovered a new class of mini microRNAs. Okay, they're only 8 bases long, and you've sequenced 5 homologs from your 5 favorite mammals. Okay, and these are the sequences that you get. Okay, and you know that they're homologous by synteny. They're in the same place in the genome, and they seem to have the same function. What could you say about their secondary structure based on this multiple alignment? You have to stare at it a little bit to see the pattern. There's a pattern here. Any ideas? Anyone have a guess at what the structure is? Yeah, go ahead.
There's a 2 base pair stem and then a 4 base loop.
Mm-hmm. 2 base pair stem, 4 base loop, and then you have the other half of the stem. So how do you know that?
So if you look at the first 2 and last 2 bases of each sequence, mm-hmm. The first and the 8th nucleotide can pair with each other, and so can the 2nd and the 7th.
Yeah. Yeah, so everyone see that. So in the first column, you have A, U, A, C, G, and that's complementary to U, A, U, G, C, right? Each base is complementary. The second position, C, A, G, G, U, complementary to G, U, C, U, A. There's one like slight exception there. Yeah, so it turns out that RNA, although the Watson-Crick pairs, GC and AU, are the most stable, GU pairs are only a little bit less stable than AU pairs, and they occur in natural RNA molecules. So GU is allowed in RNA, even though you would never see that in DNA. Okay, yeah, so everyone see that? So the structure is, well, I think I have it here. It's, you see the sort of this would be covariation. You're preserving you're changing the bases, but preserving the ability to pair. So when one base changes, when the first base changes from A to U, the last base changes from U to A in order to preserve that pairing. You wouldn't know that if you just had two sequences, but once you get several sequences, it can be pretty compelling and allow you to make a pretty strong inference that that is the structure of that molecule. Okay, so how would you do this? So imagine you had a more realistic example where you've got a non-coding RNA that's 100 or a few hundred bases long, and you might have a multiple alignment of 50 homologous sequences. You want something, you're not going to be able to see it by eye, right? You need sort of a more objective criterion. One method that's commonly used is this statistic called mutual information. Okay? So if you look in your multiple alignment, I'll just draw this here. You have any sequences. You consider every pair of columns. This is a multiple alignment, so this column, and you calculate what we're going to call, FIX. That would be the frequency of the nucleotide X in column I, so you just count how many A's, C's, G's, and T's there are. And similarly, FJ, Y for all the possible values of X and all the possible values of Y. So these are the base frequencies in each column. And then you calculate the dinucleotide frequencies X, Y at each pair of columns. Okay, so in this column you say if there's an A here and a C here, and then there's another AC down here, and there's a total of 1, 2, 3, 4, 5, 6, 7 sequences, then FAC IJ is 2 over 7. Okay, so you calculate the frequency of each dinucleotide, but these are no longer consecutive dinucleotides in the sequence necessarily. They can be at arbitrary spacing. Okay, so you calculate those, and then you throw them into this formula. Okay, and out comes a number. So what does this formula remind you of? Have you seen a similar formula before? Someone said something with entropy. Okay, yeah, go ahead.
It reminds you of the Shannon entropy formula.
Yeah, it looks like Shannon entropy, but there's a log of a ratio in there, so it's not exactly Shannon entropy. So what other formula has a log of ratio in it? Relative entropy, right? So it actually looks exactly like relative entropy. So relative entropy of what versus what? Who can sort of say more precisely if it's? We'll say it's relative entropy of something versus of P versus Q, and what is P and what is Q? Yeah, in the back.
Relative entropy of co-occurrence versus independent occurrence.
Good. Yeah, co-occurrence of F. Everyone get that? Co-occurrence of pair of nucleotides X, Y at the positions I, J versus Q is independent occurrence. So if X and Y occurred independently, they would have this frequency. Okay. So if you think about it, you calculate the frequency of each base at each column in the multiple alignment, and this is your null hypothesis. You're going to assume what if they're evolving independently, right? So any if it's not a folded RNA, or if it's a folded RNA but those two columns don't happen to interact, there's no reason to suspect that those bases would have any relationship to each other. So this is like your expected value of the frequency of xy at position ij, and then this p is your observed value. So you're taking relative entropy of basically of observed over expected, okay? And so relative entropy has, I haven't proved this, but it's non-negative. It can be 0, and then it goes up to some maximum positive value, but it's never negative. And what would it be if, in fact, p were equal to q? What would this formula give? This is, we're saying, you know, suppose this. In general, this this won't be true, but suppose it was equal to that. We've got mij equals summation of what? That log of this, which is equal to this, right? So it's fxi fy j over the same thing, log of, hope you can see that, log of 1 is 0, right? So it's just 0, okay? So if the nucleotides at the two columns occur completely independently, mutual information is 0. And that's why that's one reason it's called mutual information. There's no information, you know, knowing what's at column I gives you no information about column J. Okay, so remember, relative entropies are measures of information, not entropy. Okay, and what is the maximum value that the mutual information could have? Any ideas on that? Any guesses? That's Joe, yeah.
You could have like log base 2 of 1 over f sub x, f sub y.
Of 1, okay. So you're saying if one of the particular dinucleotides has a frequency of 1?
Yeah, yeah. So if they're like always the same.
Right.
Whenever there's like an A, there's always a T.
Yeah, so whenever there's an A, there's always a G, or a T, let's say. Yeah, okay.
So then you'd get O1 in the numerator, and like the relative like probabilities in the bottom, which would be maximized if they were all even. So
if they're all uniform. Yeah. So did everyone get that? So the maximum occurs if fxi and j, if they're both uniform, so they're a quarter for every base at both positions. Okay, sort of, that's the maximum entropy, right, in the background distribution. But then if fxy ij equals 1/4 for example, x equals y, or in our case, we're not interested in that. We're interested in x equals complement of y. C of y is going to be the complement of y. Okay. Yeah, and 0 otherwise for x not equal complement of y. Okay, so for example, if we have only the dinucleotides AT, CG, GC, and TA occur, and each of them occurs with a frequency of 1/4, then you'll have 4 terms in the sum, because remember, the 0 log 0 is 0, so you'll have 4 terms in the sum, and each of them will look like 1/4 log 1/4 over 1/4 times 1/4, right? And so this will be 4, and so log 2 of 4 is 2, and so you have 4 terms that are each 1/4 times 2, and so you'll get 2. Well, this is not a sum. I mean, this is the 4 These are the 4 terms. These are the individual non-zero terms in that sum. Does that make sense? Everyone get this? Okay. Yeah, so that's why this is a useful measure of covariation, okay? If they really, if what's in one column really strongly influences what's in the other column, and there's a lot of variation in the 2 columns, and so you can really see that covariation well, then mutual information is maximized, okay? And that's basically what we just said is written down here, okay? So yeah, it's actually, it's maximal. They don't have to be complementary, right? It would be achieve this maximum of 2 if they are complementary, but it would be also if they had some other, very specific, relationship between the nucleotides. So it doesn't. So you might want to, if you're going to use this, the way you would use it is take your multiple alignment, calculate the mutual information of each pair of columns. Okay, so you actually have to make a table, I versus j, all possible pairs of columns, and then you're going to look for like the really high values. And then when you find those high values, you want to see that when you look at what actual bases are tending to occur together, you want to see that they're actually, the bases that are complementary to one another. And another thing that you'd want to see is you'd want to see that consecutive positions in one part of the alignment are covariating with consecutive positions in another part of the alignment in the right way, in the sort of inverse complementary way that RNA likes to pair. Does that make sense? So in a sort of nested way, in your multiple alignment, if you saw that this one covaried with that, and then you also saw that the next base covaried with the base right before this one, okay, and this one covariates with that one, that starts to look like a stem, right? It's much more likely that you have a 3-base stem than that you just have some, separate, isolated base pair out in the middle of nowhere, right? It turns out it takes a few bases to make a good thermodynamically stable stem, and so you want to look for blocks of these things, okay? And so this works pretty well. Yeah, actually, one point I want to make first is that mutual information is nice because it's kind of a useful concept, and it also relates to some of the entropy and relative entropy we've been talking about in the course before. But it's not the only statistic that would work in practice. You can use it. You can use any measure of basically non-independence between distributions, like a chi-square statistic would probably work equally well in practice. Okay, and so here is a multiple alignment of a bunch of sequences, and what I've done is put boxes around columns that have significant mutual information with other sets of columns. Okay, so for example, this set of columns here at the left, at the far left, has significant mutual information with the ones at the far right, and these 4 positions covary with these 4, and so forth. So can you tell, based on looking at this pattern of covariation, what the structure is going to be? Okay, the first, let's say we start up here. The first is going to pair with the last, right? Okay, with something at the end. Then we're going to have something here in the middle that pairs with something else nearby, right? Then we have something here that pairs with something else nearby. Then we have another like that, right? Does that make sense? So there's these 3 pairs of columns in the middle, these two, and these two, and then they're surrounded by this thing, the first pairing with the last. And so it's a clover leaf, so that's tRNA. Okay. Yeah?
How would so with that previous slide, this table here, you could create a covariation matrix.
Mm-hmm.
How would that, and it could be...
How does that covariation matrix, how do you convert it to this representation? Or?
I'm just wondering how this would show up, like let's say you took the covariation matrix.
Oh, what would it look like?
Visualize it as a heat map.
In the covariation matrix?
Yeah, what would it look like in this particular example?
Yeah, that's a good question. Okay, let's do that. I haven't thought about that before, so we'll have to, have to help me on this. OK, so there would be, here's the beginning. OK, we're going to write the sequence from 1 to n in both dimensions. OK, and so here's the beginning, and it covaries with the end, right? So this first would have a covariation with the last, right? And then the second would covary with the second to last, and so forth. So you get a little diagonal down here, right? That's this top stem here, right? And then what about the second stem? So then you'd have something down here that's going to covary with something kind of nearby it, right? So that's going to be, so block 2 is going to covary with block 3. And again, it's going to be this inverse complementary kind of thing like that. Yes? Yeah. I mean, it's symmetrical, right? So you get this covering with that. But you only have to do, one half, right? So you can just do like this upper half here. Yeah, so you get that. So you'd get, I believe you get, it would look something like that.
So it would be diagonal lines orthogonal to the diagonal of the matrix.
Yeah, that's because they're inverse complementary.
Right.
Right?
Okay.
That make sense? Good question. All right, but we'll see an example like that later, actually, as it turns out. OK. All right. So here's my question for you. You're studying this non-coding RNA. It has some length. You have some number of sequences. They might have some structure. Is this method going to work for you, or is it not? What is required for it to work? For example, would I want to isolate this gene, this non-coding RNA gene, just from primates, from like, human, gorilla, chimp, orangutan, let's say, and do that alignment, or would I want to go further? Would I want to go back to the rodents and dog, horse? How far do you want to go? Yeah, question.
I think we need a very strong sequence alignment for this, so we cannot go very far. Because if we don't have a high percentage homology, then we will see all sorts of false positives.
Absolutely right. So if you go too far, your alignment will suffer, and you need an alignment in order to identify the corresponding columns. So that puts an upper limit on how far you can go, right? Okay, good, excellent point. Is there a lower limit? I mean, do you want to go as close as possible, like this example I gave with human, chimp, orangutan, or is that too close? Why is too close bad? Tim.
Maybe if they're too close, then the sequences haven't diverged enough to give you enough information about the covariation.
Yeah, exactly. If they're all the same, actually you'll get 1 times 1 over 1 in that mutual information statistic, which log of that is going to be 0. There's 0 mutual information if they're all the same. Okay, so there has to be some variation, and the structure has to be conserved. That's key, right? You have to assume that the structure is well conserved, and you have to have a good alignment, and there has to be some variation, a certain amount of variation. Those are basically the three keys. Secondary structure has to be more highly conserved than sequence, sufficient divergence so that you have these variations, and sufficient number of homologs to get good statistics, and not so far that that your alignment is bad. Sorry about that. Sally.
It seems like another thing that we assume here is that you can project it onto a plane and it'll lie flat. So if you have some very important weird folding that allows you to say crisscross the rainbow thing.
Yeah, crisscross the rainbow. Yeah, very good question. Yeah, no, that is it has to be able to so in the example of tRNA, if you were to do that arc diagram for tRNA, it would look like you'd have the big arc, that's the first and last, and then you'd have these 3 nested arcs, nothing crisscrossing, right? What if I saw, let me give you an example. 2 blocks of sequence that have a relationship like that. Is that okay? With this method, with covariation, that's okay. There's no problem. What does this structure look like? Okay, yeah, so sorry about that. So you have a stem, okay? Then you have a loop, okay? And then a stem. So this is 1 pairs with 3. That's 1, that's 3. Then you've got 2 up here, but 2 pairs with 4. So 2 pairs with, here's 4 over here. So I'm going to have, 4 is going to have to come back up here and pair with 2, okay? This is 2 over here. So that is called a pseudoknot. Okay, it's not really a knot because this thing doesn't go through the loop, and but it kind of behaves like a knot in some ways. And so, do these actually occur in natural RNAs? Yes, Tim is nodding. And are they important? Can you give an example where they are important biologically? Ribozymes. Riboswitches. Okay. We're going to come to what riboswitches are in a moment for those not familiar. Yeah, and I think I have an example later of a pseudoknot that's important. So that's a good question. Yeah, I think I should have added to this list the point that you made in the back about not being, that they have to be close enough that you can get a good alignment. That would be, yeah, I should add that to this list. Thanks. That's a good point. Okay. All right. So classes of non-coding RNAs, as promised. My favorites listed here. Everyone knows tRNAs, rRNAs. You can think of UTRs as being non-coding RNAs. They often have structure, and that can be involved in regulating the message. snRNAs involved in splicing. SNO RNAs, small nucleolar RNAs, are involved in directing modification of other RNAs, such as ribosomal RNAs and snRNAs, for example. Terminators of transcription in prokaryotes are like little stem loop structures. RNase P is an important enzyme. SRP is involved in targeting proteins with signal peptides to the export machinery. We won't go into tMRNA, microRNAs, and link RNAs you probably know, and riboswitches. Okay. So Tim, can you tell us what a riboswitch is?
A riboswitch is any RNA structure that changes conformation according to some stimulus. So that can either be some small molecule addition or something in the cell. It could be an ion, but it will change its structure.
Right. So, yeah, great. That's great. So just for those who may not have heard, or I'll just say it again. So a riboswitch is any RNA that can have multiple conformations and changes conformation in response to some stimulus, temperature, ligand, binding of some ligand, small molecule, something like that, et cetera. And often the structure, one of those structures will block a particular regulatory element. I'll show an example in a moment. And so when it's in one conformation, the gene will be repressed, and when it's in the other, it'll be on. So you can use, it's a way of using RNA secondary structure to sense what's going on in the cell and to appropriately regulate gene expression. Okay. All right. So now we're going to talk about a second approach. So this would be the approach. You've got some RNA. It may or may not do something, and maybe you can't find any homologs. It might be some newly evolved species-specific RNA, or you're studying some obscure species where you don't have a lot of genomic sequence around. So you want to use the first principles approach, the energy minimization approach. Or maybe you have the homologs, but the alignment, you don't trust your alignment. You want a second opinion on what the structure is going to be. So just in the way that protein folding is, you can think of an equilibrium model where it's determined by folding free energy, and enthalpy will favor base pairing. You gain some enthalpy when you form a hydrogen bond, and entropy will tend to favor unfolding, right? So an RNA molecule that's linear has all this conformational flexibility, and you lose some of that when you form a stem, right? It forms a helix. Those things don't have as much flexibility, and even the nucleotides in the loop are a little bit conformationally. They're not as flexible as they were when it was linear. Okay, so that means that at high temperatures it'll favor unfolding. Okay, so the earliest approaches were approaches that sought to maximize the number of base pairs. Right, so you gain some. So they basically ignore entropy and focus on the enthalpy that you gain from forming base pairs. And so Ruth Nusinoff described the first algorithm to figure out what is the maximum number of base pairs that you can form in an RNA, okay? And so a way to think about this is, imagine you've got this sequence. What is the largest number of base pairs I can form with this sequence? You just draw all the possible base pairs. That A could pair with that T. This A could pair with that T. They can't both pair simultaneously, right? And this C could pair with that G. So if we don't allow crossing, which now we're going to, like coming back to Sally's point, we're not going this would cross this, right? So we're not going to allow that. So the best you could do would be to have this A pair with this C and this C pair with this G and form this little structure. This is not realistic because RNA loops are not one, it can't be one base. The minimum is about 3. But just for the sake of argument, okay? You can list all these out. But imagine now you've got 100 bases here, right? every base will on average potentially be able to pair with 24 or 25 other bases, right? So you're just going to have just an incredible mishmash of possible lines all crisscrossing, right? So how do you figure out which, how to maximize those that pairing? Any ideas? Yeah.
You look for sections of homology. And so
we're not using homology. We're ab initio.
Sorry, not homology, but like sections where complementarity there.
Okay.
That's what I was
so blocks of complementarity.
Yeah, and then so.
You could blast the sequence against the inverse complement of itself and look for little blocks. You could do that. Yeah. That's not what people generally do, mostly because the blocks of complementarity in real RNA structures are really short. They can be 2, 3, 4 bases. Sally, yeah.
Could you use some kind of dynamic programming approach where you just start with a very small case and build up?
Mm-hmm. Yeah, so we've seen that work for protein sequence alignment. We've seen it work for the Viterbi algorithm. So that is sort of the go-to approach in bioinformatics, is to use some sort of dynamic programming. Now this one, for RNA secondary structure that Nusanov came up with, is a little bit different than the others, okay? So you'll see it has a kind of different flavor. It turns out to be actually, it's a little harder to get your head around at the beginning, but it's actually easier to do by hand, okay? So let's take a look at that. Okay, so recursive maximization of base pairing. Now, the thing about base pairing that's different from these other problems is that the first base in the sequence can base pair with the last, right? There's nothing, how do you chop up the sequence? Remember with Needleman-Wunsch and with Viterbi, we go from we go from the beginning to the end, and that's a logical order. But with base pairing, that's actually not a logical order. You can't really do it that way, okay? So instead, you go from the inside out. You start in the middle of the sequence, okay, and work your way outwards in both directions, okay? Or another way to think about it is you start with you write the sequence from 1 to n on both axes, and then we'll see that we initiate the diagonal all to zeros, and then we think about these positions here next. So 1 versus 2, we consider, could 1 pair with 2? And could 2 pair with 3? Those are like little bits of possible RNA secondary structure. Again, we're ignoring this fact that loops have to be a certain minimum length. This is sort of a simplified case. And then you build outward. So you conclude that, okay, base 4 here could pair with base 5. Okay, so we're going to put a 1 there. And then we're going to build outward from that toward the beginning of the sequence and toward the end, adding additional base pairs when we can. Okay, that's basically the way the algorithm works. And so the key idea That's one key idea, that we go from sort of close sequences, work outward to far away sequences. And the second key idea is that the relationship that, like, as you add more bases on the outside of what you've already got, that the optimal structure in that larger portion of sequence space is related to the optimal structures of smaller portions of it in one of 4 different ways. And these are the 4 ways. Okay, so yeah, let's look at these. So yeah, so the first one is probably the simplest, where if you've got, you're doing this, you're here somewhere, meaning you've compared sequences from position, let's say, I minus 1 to j minus 1 here. And then we're going to consider adding, like, actually, it depends how you number your sequence. So let me see, how is this done? Yeah, sorry, I plus 1. Yeah. I plus 1 to j minus 1. Okay, we figured out what the optimal structure is in here, let's suppose. Okay, and now we're going to consider adding one more base on either end. We're going to add j down here. Okay, and we're going to ask if it pairs with I. Okay, and if so, we're going to take whatever the optimal structure was in here, and we're going to add one base pair, and we're going to add plus 1 because now it's got one additional. We're counting base pairs. Okay, so that's that first case there. And then the second case is you could also consider just adding one unpaired base onto whatever structure you had, and then you don't add one. And that could be on, you can go in either direction. You can go sort of toward the beginning of the sequence or toward the end of the sequence. And then the third one is the tricky one, is this what's called a bifurcation. You could consider that actually I and j are both paired, but not with each other. Okay, that I pairs with something that was inside here, okay, and j pairs with something that was inside here. So your optimal parse from I to j, if you will, is not going to come from the optimal parse from I plus 1 to j minus 1. It's going to come from, like, rethinking this and doing the optimal parse from here to here and from here to here and combining those two. Okay, so it's probably confused by now. So let me try to do an example. And then I have an analogy that will confuse you further. So wait, ask me for that one. Okay. This was the simplest one I could come up with that had this property. Okay, so we said before that if you were doing the optimal from 1 to 5, that it would be the AC pairing with the GT, right? We did that one, okay? And now, if you notice, this guy is kind of a similar sequence, right? I just added a T at the beginning and an A at the end. And so you can probably imagine that the best structure of this is here, right? Those 3, you get 3 pairs of this subsequence here. Okay, I mean that's as good as you can do with 7 bases. You can only get 3 pairs. So, and this is as good as you can do with 5. So these are clearly optimal. Okay, so the issue comes that if you're starting from somewhere in the middle here, let's say you're, let's see, how would you be doing this? I don't know. You start here. Let's suppose the first 2 you consider are these 2. You consider pairing that T with that A. You can see this is not gonna go well, right? You might end up with that as your optimal substructure of this region. Remember, you're working from the inside out. So you're going from here to here, and you end up with that. Okay. Now, and then you add, what do you do here? You don't have a G to pair the C to, so you add another unpaired base. Now you've got this optimal substructure of the sequence that's almost the whole sequence. It's just missing the first and last bases, but it only has 3 base pairs, right? So when you go to add this, you can say, oh, I can't add any more base pairs, so I've only got 3. But you should consider that we already solved the optimal structure of that, and we had 2 nice pairs here, right? We had that pair, and we already solved the substructure of the optimal structure of this portion here, and we had those 3 pairs. And so you can combine those 2, and all of a sudden you can do much better. Okay, so that's what that bifurcation thing is about. Okay. All right, so this is the recursion working out. You can see that's the base pairing one. You add 1, or you can just add an unpaired base and you don't add anything, or you consider all the possible locations of bifurcations in between the 2 positions that you're adding, I and j, and you consider all the possible pairs, and you just sum up each pair and go, I'm sorry, you don't sum them up, you consider them all, and then you take the maximum. Okay. All right, so the algorithm is to take an n by n matrix, initialize the diagonal to 0, and initialize the subdiagonal to 0 also. Just don't think too much about that, just do it, and then fill in this matrix recursively from the diagonal up and to the right. And it actually doesn't matter what order you fill it in, as long as you're kind of working your way up and to the right. You have to have the thing to the left and the thing below already filled in if you're going to fill in a box. Okay? And then you keep track of the optimal score, which is going to be the sum of base pairs, and then you also keep track of how you got there. What base pair did you add, so that you can trace back. And then when you get up to the upper right corner of this matrix, you then trace back. Okay? So here is a partially filled in matrix. This is from the Nature Biotechnology review. And the zeros are filled in. So here's what I want you to do at home, is print out or photocopy or whatever, make this matrix, or make a bigger version of it perhaps, and look at the sequence and fill in this matrix. And fill in the little arrows every time you add a base pair. It's actually not that hard. There are no bifurcations in this, so you can ignore. That's the tricky one. Ignore that one. You'll just be adding base pairs. It'll be pretty easy. And then you can reconstruct the sequence. So here it is filled in, and the answer is given, so you can check yourself. But do it without looking at the answer. And then you go to the upper right corner. That means it's the optimal structure from the beginning of the sequence to the end, which of course was our goal all along. And then you trace back, and you can see whenever you're moving diagonally here, you're adding a base pair. Okay, diagonal. Remember, you add one on each end, and so you're moving diagonally and adding a base pair, and you get this little structure here. Okay. All right. Okay. So computational complexity of the algorithm. You can think about this, but I'll just tell you it's memory n squared, because you got to fill in this matrix. It's the square of the length of the sequence. Time n cubed. Okay, this is bad now. Okay, and why is it n cubed? It's n cubed because you have to fill in a matrix that's n by n, and then when you do that maximization step, that check for bifurcations, that's sort of order n as well. Okay, so n cubed. So this means that RNA folding is slow, and in fact, some of the servers won't allow you to fold anything more than like 1,000 bases, because they'll take forever or something like that. So, and it cannot handle pseudoknots. If you think through the recursion, pseudoknots will be a problem. Okay, I'm going to just show you. I want to show you. I'll get to this. That these are from viruses. Real viruses have some of them have pseudoknots, like these ones shown here. And some even have these kissing loops, which is another type where the loops, the two stem loops, the loops interact. And the pseudoknots in particular are important in the viral life cycle. They can actually cause programmed ribosomal frame shifting. Okay, they cause, when the ribosome hits one of these things, normally it just denatures RNA secondary structure. When it hits a pseudoknot, it'll actually get like knocked back by 1 and will start translating in a different frame. And that's actually useful to the virus to do that under certain circumstances. That's how HIV makes the replicative polymerase, is by doing a frame shift on the ribosome, okay, using a pseudoknot. So these things are important. And I just had well, I won't anyway. There's fancier methods that use more sophisticated thermodynamic models where GC counts more than AU, And I won't go into the details, but I just want to show you some pretty pictures here that the Zucker algorithm is this is a real-world RNA folding algorithm, calculates not only the minimum energy fold, but also suboptimal folds and the probabilities of particular base pairs, summing over all the possible structures that RNA could form, weighted by their free energy. Okay? So it's the full partition function. It's not perfectly accurate. It gets about 70% of base pairs correct, which means it usually gets things right, but occasionally totally wrong. And there's a website for the mFold server, which is actually one of the most beautiful websites in bioinformatics, I would say. And there's also, if you want to run it locally, you should download the Vienna RNA fold package, which has a very similar algorithm. And I just want to show you one or two examples. So this is the U5 snRNA. This is the output of mFold. It predicts this structure. And then this is what's called the energy dot plot, which shows the bases in the optimal structure down below here, and then in sort of these suboptimal structures here. And you can see there's no ambiguity. It's totally confident in this structure. Then I ran the lysine riboswitch through this program, and I got this. I got the minimum free energy structure down in the lower left, and then you see there's a lot of other colored dots. Okay, those are from the suboptimal structures. So it looks like this thing has multiple structures, okay, which, of course, it does. So the way that this one works is it can form, in the absence of lysine, it forms this structure where the ribosome binding sequence, this is prokaryotic, is exposed. And so the ribosome can enter and translate these lysine biosynthetic enzymes. But then when lysine accumulates to a certain level, it can interact with the RNA and shift its structure so that you now form this stem which sequesters the ribosome binding sequence and blocks lysine biosynthesis. So a very clever system. And it turns out that quite a few, there's like dozens of these things in bacterial genomes, and they control a lot of metabolism. So they're very important. And there may be some in eukaryotes too, and that would be good. If anyone's looking for a project, not happy with their current project, you might think about looking for more riboswitches. So I'm going to have to end there, and thank you guys for your attention, and good luck on the midterm.